Welcome everybody to Chem 1A. Yeah. How's everybody doing? Wonderful move-in and a wonderful week, nice sunny week. I'm glad you're all able to find us since we're at the center of the world. Yeah, this is Berkeley. So we're, as you've noticed, this is a pretty large class. There are 3 lecture sections of this size, but you're all divided into groups of about 30 students, and there's plenty of support, and we try to be as personable as we can through B-Space communications and getting you into lab sections, and we have plenty of office hours and help for you to study and learn the material. So don't be too alarmed for those of you who are new to Berkeley about the size of the class. So to begin, what is the course about? Well, this slide, as this slide indicates, we're going to start at the beginning. We consider chemistry to be the center of science, the beginning of science. We hope that in this course, because for many of you it's a prerequisite for going on in engineering or the life sciences, this course is about laying a foundation for you. And I want to say right at the outset that this is different than for many of you who have just come from high school. This is different than what you did in high school, where you memorized for a course, did really well in the exam, and then don't remember anything, okay? We want to start to build skills. This is a course where you're going to need to really understand and be able to use the material going forward for the next many years as you go on in science, all right? So be aware that you really need to start thinking differently about how you study. So what is chemistry? These are some web definitions of chemistry. The science of matter and the changes that it undergoes. So basically it's about everything. Everything around you is matter. And we're going to study it, and we're going to think about it, and we're going to apply it to your everyday experience. Another definition is the composition, structure, properties, and reactions of matter, especially at the atomic and molecular level. And then there's also this definition of mutual attraction or sympathy, rapport. Yeah, that's one use of chemistry. We're not going to get into that unless you want to personify atoms and talk about their mutual attractions and the sympathy that they have for one another. And that being said, the key component of this course is to develop an atomic view of the world. When you walk out of here, you're not going to be looking at the world in the same way, we hope, because you're going to have this image of these little atoms and what they might be doing in the materials and that are all around you. All right, now, chemicals end up in the news a lot. What I want to emphasize is that the word chemical refers to everything, not just the things that you hear in the news, which often put a bad light on chemicals. In fact, chemicals can be quite good. I can give you examples in just about any area, from pharmaceuticals that really help you to the great materials that we have that we use every day. And what I've done here is to emphasize that we've divided the course into 4 sections. Unit 1 is on matter, Unit 2 is on change, Unit 3 is on energy, and Unit 4 is light and matter. And I put up an example here which is related to what I might call green chemistry, or the environment, which will be an underlying theme that goes through the semester, especially in the laboratory sections where you're going to be thinking about this. And so the examples I show you here is, how do you think about matter? In this case, what are the properties of this bottle cap such that when you dispose of it, it actually degrades over time rather than filling up a large landfill? These solutions here are called green works. They are cleaning solutions that don't contaminate the environment. They cause reactions to get soils and stains and remove grease, etc. And so you might think about what chemicals are in there and how does that work. The third unit on energy, I have a bus here that's hydrogen powered. We have some electric buses running around the area, and you might think about what chemistry went in to make that happen. And finally, light interacts with matter. We'll end with that as the fourth unit. And I have a solar panel here. So what kind of matter, what kind of materials do you need in order to take all that energy that's coming from the sun and power your home? All right, so the course outline, as I said, we go from matter to change to energy and then to light and matter, the interaction of light and matter as a capstone. And so for the first section, we're going to be looking at the properties of matter, and we're going to try to understand what are the atoms, ions, and molecules doing in order to give matter those properties. All right, so now for some introductions. I'm Professor Stacy. I am in charge of the 9 a.m. lecture, this lecture section. We have 3 other professors who are involved with the course. And I'd like to introduce them just so that you know all of us. And I want to emphasize that we are all available to you, to any questions you might have. So the 11 a.m. section is taught by Professor Maida. Stand up. And for those of you who, he's often at the 9 a.m. lecture because he likes to listen to what I do and then give me feedback. Sometimes if I make an error, you'll see him kind of rolling with laughter on the floor over there as I look over to him. But what we're going to do in class, as you'll see with the eye clickers, is do a lot of discussion. So there's a real advantage to kind of hanging out in that area because you have his expertise sitting there. The 1 p.m. lecture is taught by Professor John Arnold. There's John. And same thing, John's often in class, and it's a real advantage, you know, to place yourself so that you can get the benefit of his wisdom. The labs are coordinated by Dr. Dusky, and she's up here. And Dr. Dusky is wonderful in terms of taking care of the teaching assistants that will be in your labs and also taking care of you as issues arise for with just mechanical difficulties and whatnot that you might have. The lecture demos, Karen's disappeared, but she'll be back later. The one thing that you're, oh, there she is, Karen Chan. And the one thing that people will come back 10 years from now, and you know what they'll remember about Berkeley? The lecture demos in Chem 1A. You won't remember me, won't remember the rest of us, but they will remember Karen and the wonderful demos that she sets up. So you'll see those. And each of you have a graduate student instructor that will be assigned to your laboratory section. So they will do a discussion section with you and a laboratory section, and will be available for office hours. These are all graduate students, largely in the chemistry department. They're all working towards their PhD, and they're very knowledgeable and really are able to present, really to support the students in the class. So if there are any graduate student instructors that in the room, could you just please stand up so that we can see who you are? Okay. And so they're also spread around the room and often at lecture. They'll be at one of the 3 lecture sections. So please use them as well in order to follow what's going on. And for those of you who haven't already gotten there, BSpace is an important website for you. It's where all the course materials are. Okay? And a lot of information. We have questions and answers on there. A lot of information is on there. The one last thing that I want to say is the 3 of us, Professor Maida, Professor Arnold, and myself, our role here at the university is to teach undergraduate courses, but also to do research with graduate students and with undergraduates. So as the semester goes on, I hope we'll be able to share with you some of the research that we do, and I hope that over time, as you get familiar with Berkeley, that you will also participate in research in one of the departments here. That is really the big benefit, big bonus of being here at Berkeley, is the fabulous things that you can get involved in. So please consider that as time goes on. All right, so now to begin with, we're going to practice with your eye clicker. Now, if everyone doesn't have it, don't worry about it. If you can't read your code, come see me after class. We'll figure out what the code is that rubbed off. But for those of you who have it, take it out. All right, turn it on. Should have a blue light. All right, now nothing's going to happen until I start the iClicker program. All right, so this is an opportunity for you to introduce yourselves to the entire class. And the first question I have is, what is your major or intended major? Now, obviously, there's no right answer here, okay? So don't worry about that. So I go up here, and I start it, and it will then, it's set a timer for 2 minutes. You can see it counting down. And those numbers that are going up there are the people in the classroom who are actually registering a vote. Now, if you registered a vote and you don't like it and you want to change it, you can change it, all right, until the time runs out or until I stop it. All right, does that make sense? Any questions on that? All right, so do we have, people are getting there, numbers are going up, you have to wait till they stop. Yeah? So how many students are in this section is a question. The room seats about 550. Okay? All right, so are we almost done?
How do you know which iClicker is us?
The question is, how do I know which iClicker is you? Because you've gone online to register. Okay, if you haven't gone online to register, your vote won't get registered right now. Okay? So you have to go online and you put in your student ID number and you put in your iClicker number. Okay, it's all linked to the B-Space. iClicker.com. And it's okay for today. We're just getting to know each other. So I'm going to stop this now, and what I can then do is display the votes. All right. And so what you see is a distribution for those of you who have iClickers. We have about 60% of the students in the class are considering a life science major, and about 14% are engineering, and we have a number who are other science, a number who are non-science majors, and a number who are undecided. And so as we go through the course, we're going to use iClicker each day to kind of check in with you. Now, we're going to give you 2 points per day when we do this. One for just being here and participating. One for getting one of the questions right. We're not trying to trick you on these questions. What we're really trying to do is just be able to check in with you. If nobody understood something, I'm going to go over it again. And so this is a way to just break up the lecture and check on your understanding. Okay? Now, I have one more iClicker question for you. Where did you attend high school? There is a right answer here. And usually while this is going on, there is a lot of chatter, and that's fine. This is a great time for you to just say hello to the people sitting next to you, okay? Okay, so let's take a look. Let's take a quick look at what this looks like. All right. So you see the distribution there. There's quite a number of you who have not left the Bay Area. We have a fair number who've come up from the Los Angeles area and other places in California. Other states and foreign countries are a smaller number. And so what I would do is invite those of you who are from California and especially from the Bay Area, to get to know those students, to welcome them, to teach them a little bit about the culture around here. Those are good people to start to get to know. You can show them around, and they hella need to understand how to speak from the Bay Area. All right. And so the right answer here is A. All right. All right, so now the course materials, just quickly, I want to go over. Most of you, this is all posted on BSpace. There are emails to contact us, chem1af10@gmail.com. If you have any questions, please email. If you have iClicker problems, there's an iClicker, specific iClicker email. That's all on BSpace. You need to get the AL ebook access code. The AL is a homework system. You need the access code to get into the homework system, and you will have homework assignments each week. And it is linked to an electronic book that you can use online if you choose. The discussion manual is a specific slim reader that you have to get to join in on the discussions. That's at NEDS. Laboratory manual, a laboratory notebook with carbon copy pages. We don't care what it looks like, but you need to be able to hand in, when you're taking notes in lab, you need to be able to hand in the carbon copy. iClicker, you see what we're using it for. When we say scientific calculator, simple one, we don't care which one you use, but when you come to a midterm, we don't want you to bring in a computer with you. You know, some of these calculators you could store the whole book on, and we won't allow you to come in with one of those. And as far as, we'll say much more about this, but as far as the textbook goes, they're all the same. We don't care. Choose one that you like. If you don't like the ebook one, get another one. It's optional, but you're going to need resources. You can look on the web. You can learn things from the web. You can also learn them from the book. So there was a question here. Is the AL access code the one that you got for free with the textbook? Oh, you need to buy the AL ebook combo together, right, so that you get the Berkeley one. And by the way, for those of you who haven't done it yet, there was it's kind of an optional assignment, but it was a way to get you started and a way for us to get to know you and support some of you who feel like you have weaker backgrounds. We can support you if you go into AL and take that diagnostic assignment that we gave you. So about half of you have already done that. So I would really encourage the rest of you to get there and do that. All right? It'll really help you get to the first assignment and whatnot. Any other quick questions on this? All right, and again, just email if there's any issues that you might have. I don't want to spend too much time on the policies. There's a whole document of policies. We're going to have 4 midterms and a final exam. Because of the size of the class, there are no makeups. So if you cannot attend these exams, at the time that they're scheduled, you should not be in the class. Now, that being said, there are exceptions for athletes. And obviously, there are exceptions for bona fide excuses, such as being ill or some other personal issue that might have arisen. But you need to make sure the dates are all posted that you can attend these midterms and the final. You also have to attend all the laboratory periods. Again, you cannot, there are no excused, one excused absence, unexcused we'll deal with. But really, you have to be at these laboratory sessions. And we'll say more about how the points are distributed. They're largely based on the midterms, which do include questions on the labs. You'll find that we are giving these chem quizzes on iClicker during lecture. There are discussion quizzes at the end of discussion. Lab reports are due at the beginning of the discussion, but your laboratory GSI or graduate student assistant will go over this with you. The homework is due Wednesday by 9 a.m. You can make many, many attempts on the online homework. until you get it right, and then you can submit it before Wednesday at 9 a.m. For those of you who have lab today, some of you already had lab yesterday, we do require that you're wearing leg covering and closed-toed shoes. And it's not a good idea to show up in shorts and flip-flops because the stockroom will offer you a pair of weird baggy white pants and covering to go over your foot, and you will be remembered as the person wearing that for a long time to come. So it's not a good idea. Cheating policies are strictly enforced. All right, we are on top of you. And I want to say a couple things about what you're doing in the lecture. Laptops are okay. I would like them to be over to the sides and not so that they're not disturbing everyone else. And if you're coming to class and you're on your Facebook site and you're playing games, don't bother to come to class. All right, now, the point I want to make is my friends up in the booth can actually see everything that's going on. And right now, they will put the camera on the audience. All right? All right, so I'd say somebody ought to close their laptop, Dr. Dusky. And they can move around. It's like, at the football game, they can zoom in on you. All right, this is kind of like big brother, big sister up there. But my point is we really want you to do well in the course. We want you to do your own work. We catch people coming with 2 eye clickers while their friend is in bed, all right? Don't do that. All right, enough said. All right, the grading scale is absolute, meaning we would be happy to give every single person in this class an A. And you're all capable. I don't care how much chemistry you've had before. We know that people who've had very little chemistry get an A. We also know that people who've got a lot of chemistry don't get an A necessarily. So this is a kind of a typical grading distribution. And remember, you guys were all the A+ students in your high school. So how do we get to this kind of grade distribution? If everyone can get an A, why doesn't everyone get an A? And so I have one more eye clicker question for you. What do you think are the behaviors of an A student? All right, let's see what you think. Stop it, display. All right. There's a few of you who need to get clued in here, but so let me just emphasize a couple of points here. You need, first of all, I posted the slides for today's lecture on B-Space. I don't care whether you have them today or not. But my point is, I will always post the slides. You can print them out, but when you come in, you can't just be sitting there going, yeah. Take notes. You're not going to remember all the details. There's a lot of questions on the slides. Answer those questions during lecture. Take notes. We're going to not require a specific textbook, but I will post a study guide, and that study guide will tell you, go out and read things, look at problems. Don't just memorize. You need to go for understanding. By the way, this is step by step. The worst thing, you know those people who came in and thought they knew everything? You know what happened to them on the first midterm when they thought they could cram 2 days before the midterm? Didn't work. Why not? Because the lectures build. If you don't go home and understand what we did today, you're going to have trouble understanding what we're doing on Monday. You need to very systematically, for shorter periods of time, practice after each lecture. The other common thing for you to do, because all you had to do in high school often was memorize problems, is not to work the problem for yourself. And guess what? When you see the answer, it always looks easy. All right? But on those exams, you're not going to see the answer, and it's not going to be a problem you've seen before. So you need to learn how to work them out. We offer a lot of office hours. We're going to start to get study groups together. Now, for those of you who've never worked with someone else, let me tell you, you've got a lot of smart colleagues in the classroom. And listening to their questions can really help you out. And explaining your thinking to them, that's critical. So you need to do a combination of working alone, but also working with other people. All right. So that being said, let's move to the first section. Now, I did post this part of the lecture, so some of you already have the slides printed out. This is a shorter lecture because we had the introductory material, but what it's called is fine art, representing molecules. So the first thing we're going to do is take a look at how we think about molecules, how we represent them. Okay? All right, so this begins, and by the way, there was also a pre-class assignment, and I'm not going to collect those pre-class assignments, but let me tell you, if you've looked at them, it's not hard. It's what do you think? You don't have to read anywhere. Do them, because when you come into class then, things will make a whole lot more sense. And so as you'll see, we're actually going to be talking about a lot of the things that were on the pre-class assignment. All right, so to begin with, chemistry is all about molecules. And what I want you to start doing is looking at the patterns and thinking about representations and what they're trying to tell you. So what I have here is the molecular formula for water. So what does that mean? Why would I write it as H sub 2O? What does it mean? Anybody? Go ahead. 2 hydrogens for every 1 oxygen. And you know what? They don't come apart very easily. That's why it's written H2O. All right? All right, let's go to the structural formula. What has this, oops, sorry. What is this adding to your understanding? Go ahead. All right, so that the hydrogen is single bonded to the oxygen. So there is some kind of connection, correct, between hydrogen and oxygen. What would the other possibility be? Is there another possibility? Someone in the back. I'm going to be walking around soon with my... So what would the other possibility be? Is this the only way that 2 hydrogens and an oxygen could come together? Go ahead. On this case, they're as far apart from each other as possible. I'm going to see if this will work. So can we switch over to this one? It's mine. Okay. So, but what else is going on there? They're as far apart. Is there another possibility? If I have 2 hydrogens and an oxygen, I connected them with the oxygen in the middle. Go ahead. Let's talk louder. So the oxygen's always in the middle, but is there another possibility? People, I mean, in principle, if you don't know anything, what's the other possibility? That there's a hydrogen in the middle, right? So this structural formula is telling you something else, right? It's telling you that the way they're connected. Yeah, I think I'm still not connected. I am? Practicing this. Okay. Oh, yeah, there's my little cursor. All right. What does the ball and stick model tell you? Go ahead. So someone said the size of oxygen compared to hydrogen. What else? Go ahead. You? Mm-hmm. It tells me something about the shape of the molecule. What else? Go ahead. Yeah, so there's an angle between the hydrogen atoms. What else? Is there an assumption that's being made up there? I mean, think carefully. Go ahead. It's showing that oxygen's a lot bigger than hydrogen. What is it showing about atoms? How is that visualization thinking about atoms? What are they? They're spheres. Is that correct? Should they be boxes? So you might think about what is implied by this representation. What do you think the sticks are? So bonds, some kind of way that they're put together. All right, now what about this model here, this one here, the space-filling model? What is that showing? Go ahead.
Relative size.
So it's showing, kind of trying to show relative size again, but what else? Go ahead. Yeah, so she said it's a more accurate representation because it's showing how the atoms actually are overlapping with one another, way up there. Gives a representation of bond length. All right, now here's a question. What does it not tell you? It's important to know this, by the way. And science is not just important to know what it is, but what is it not telling you? I hope somebody says, go ahead. The kind of bond? What else is it not telling you? Go ahead. What kind of atoms those are? In fact, it's color-coded way up there. Okay, it doesn't tell you anything about, say it again, the interactions. What else doesn't it tell you? Way up there? So it's not telling you anything about how that molecule might interact with other molecules. There's something really obvious here. The size. Is it that big? All right, so if I have a let's actually just look at the size here. So those distances are 3.7 angstroms. How small is that? Tiny. All right, now let me give you an idea how tiny. A glass of water, how many molecules are in a glass of water? Just estimate. 3 zeros, 4 zeros, 5 zeros, how many? I got 24 up here, I got 12 over there. How many is it? 23 zeros. All right? That's a lot of zeros. There are that many molecules. So many molecules that you can't even, there are more molecules in that glass of water than there are stars in the universe. Okay? Pretty staggering number. So how tiny are these things? Really, really tiny. And what's this last thing showing you over here with the cluster? Go ahead. Yeah, the position of the molecules and how they interact. What do you notice in a glass of water? Well, go ahead. Yeah, so whether you know the word hydrogen bond or not, do you notice that there are white ones that seem to point towards red ones, that they kind of orient themselves a bit? But are they fixed in place? Does it look like they have a fixed location? Go ahead. Yeah, so you know it's a liquid because they're not in fixed places, all right? So there's a lot to see. Even if you've seen the water molecule before, there's a lot to see. Okay, and now this is just another example. This is the glucose molecule. Glucose is one of the sugars that your body uses. We'll talk about that in a moment. So what's happened here? What's the molecular formula? So you see how many atoms are involved. Why is that not written CH2O? Why don't I just reduce it down? I can divide by 6, right? What does C6H12O6 tell you? That's the number that are in the molecule. And do molecules break apart easily? No. So those are stuck there. What's the structural formulas? You notice there are 2 up there. So what do you see here? This one shows you how they're put together. And then what's implied on this little spot here? Carbon is implied, but there's also some hydrogen atoms that are implied too. Okay? So you might take a look at how this formula and this structural formula, you might try, when you get home, to draw in the rest of the atoms here. It would really help you specifically to draw in the rest of the atoms. So I think if I can write, oops, no, never mind. Got to get my little felt tip pen. All right, see if that works. So there's a C here, and there's also another H. So you might try to do that. Oh, that wasn't very nicely drawn. Okay, I'll get used to this. All right, and then you see the ball and stick model. And then what does the space-filling model show you in this case? What's the overall shape of that molecule? Almost looks spherical, right? You get fooled when you're looking at the structural formula. This looks flat. This looks pretty flat, whereas when you get to here, it really does look ball-shaped. All right, so, sorry, I'm over here. All right, so I asked you to look at the sugars. Now, for those of you who are maybe a little bit concerned, I'm not looking for words that you might have learned in high school. I'm really looking for you to take a look here and realize that the names mean something, the formulas mean something. These molecules have some similarities. And so what I'd like to know is what do you see? What patterns show up here? Go ahead. All of them have CH2OH. Do you see that? Okay, what else? Go ahead. So does everyone see the ring of carbons? Is it just a ring of carbon? No, it always has an oxygen in there. Go ahead. Sorry, what? Oh, yeah, does everyone see that? What do they end in? OSE. So that must mean something about those molecules, right? The name is meaning something here. We're not asking you to memorize names, but we're asking you to recognize when the names are similar. Go ahead, did you have something? So the OH, does everyone see that there are lots of these OHs on there? All right, in fact, if you look around the ring, there's quite a number. What about the formulas? Way up there? Yeah, do you all notice, look at the number of carbons to hydrogens. It's 1 to 2 in the top two, but not in the other one, right? But it's close. Hydrogen to oxygen is 1 to 2. Carbon to hydrogen is 1 to 2 in the first and the third, right? Here's another thing. What do you notice about this formula and this formula? So if all you knew is the formula, would you know which molecule you had? No. And so what's different about them? Their structures. Okay? Any other comments on this one, on these? Any other things you see? Go ahead. They all contain only single bonds. What do you mean by that? I mean, if you didn't know what a bond was, what are you really saying? There's only one line. Okay? So there's only one line that's represented. Go ahead. They're all composed purely of carbon, hydrogen, and oxygen. And what you're going to notice is that there's an incredible variety of molecules that are composed of just carbon, hydrogen, and oxygen. In fact, the next course that many of you are going to take is called organic chemistry. It's not the word organic that's used, by the food people. Organic means carbon, hydrogen, and oxygen molecules. All right. So what about the neurotransmitters here? Oh, by the way, I just want to say something before I go on. So these all have very similar functions. The first thing that you might notice, if you were to taste any of these, we don't recommend you tasting things unless they're food approved, but if you did, they would all taste sweet. So there is a detector on the tip of your tongue, which is also a molecule called a receptor site, that when the sugar or when one of these molecules binds to it, sends a signal to your brain, and your brain goes, sweet. All right? glucose, as I said, is what your body breaks down from carbohydrates. Carbohydrates are just long chains of glucose molecules. Your body breaks it down and then uses the glucose molecules for energy. And we'll talk about how that happens later in the course. Sucrose is table sugar. Fructose is the fruit sugar. Okay? All right. So now, what about the neurotransmitters? Go ahead. Yeah, go ahead. They all have benzene rings. What do you mean by that? Because not everybody in the class knows what a benzene ring is. Okay. So what she's noticing is that these molecules all have this ring structure, which is interesting, right? Because do you see that it has a double, 2 lines, single line, 2 lines, single line, 2 lines. And what else? Go ahead. They all have an amine group. What do you mean by that? So NH, so here, do you see that this has a nitrogen, and a nitrogen? Do you see where the nitrogen atoms are? All right, so they all have nitrogen atoms. He called it an amine group. Where else do you see kind of that ending name? But what about those names? Dopamine, adrenaline, spelled different, pronounced differently in the English language. Okay, and methamphetamine, right? So do you see there's amine in, or that kind of word in there? All right, what else do you notice? Go ahead. They all have similar bond angles, and which ones in particular are you noticing? Yeah, so right in here, you're noticing that there's a certain bond angle, for example. Oh, sorry. The dark. Oh, and do you notice these triangles and these? What are they? What do you think that's trying to show there? We'll get more into that. Go ahead. Yeah, they're trying to show it's not flat. Trying to show that one is pointing out at you and one is going back. What else do you notice? What about the overall shapes of those molecules? Do they look same or different? You see some similarities? What do you see on the shapes? Look at this part of the molecule here. Okay, that's actually flat. And then what do you see hanging off of it? Okay, and your body is detecting these all similarly. In fact, these are some molecules that are directly implicated in getting nerve signals. So these molecules are released, they go into the end of into a nerve ending, and then there's a signal that's sent to your brain. Dopamine is an important molecule. Those who have Parkinson's disease are deficient in dopamine, and so we've learned how to administer a drug that will actually get into the brain, create dopamine, so that those individuals have reduced symptoms. Adrenaline, it's another one where when you're afraid, and you know the tingling response that you get, if you're really afraid of something or ready to fight or ready to run away, adrenaline is released in large quantities, and the signals go to your brain. And what's fascinating about this neurotransmitter is it shuts everything down except for kind of blood or whatever going to your muscles so that you can react very quickly. And then methamphetamine I wouldn't know about. Okay. Now, here are the amino acids. Tell me a little bit about the amino acids. Why are they called amino acids? What do they have that's similar? Go ahead.
They have an amine group and a carboxyl group, a hydrogen, and a side group.
Okay, so let's review that a little more carefully. So what are the two main features? You said amine group or amine group. What do you mean by that? What are the elements that are involved?
The elements are involved are nitrogen and hydrogen.
Okay, so you see each of these has this nitrogen and hydrogen group. And what else seems to be common to all of them? Go ahead. Okay, so do you see this part of it also is always there? See two bonds to, or two lines to O and one line to another O, and each of these have these. So if you look carefully, you'll see that they're all there. And then there is off of this carbon, this one right here, this one right here, there's another thing that's another two things that are hanging off of it. Okay? What about the chemical formulas here? What do you notice that you might not have, we didn't point out on the last ones? So let me just go back to, what do you see about the numbers here of hydrogen? They're even numbers, right? Look at what happens here. What happens here when you put a nitrogen in? It's odd, right? What happens here when you put a nitrogen in? Well, if you put one nitrogen in, what happens? Odd. If you put two nitrogens in, what happens? So there's for those of you who've seen a lot of this before, there's a wealth of information that you probably haven't thought about. So you need to keep looking. For those of you who haven't seen it before, just keep looking. There's a lot to see. And these amino acids, of course, are really important because they're able to link up and create the protein molecules. So my hair, my skin, my fingernails are all just long chains of these kinds of molecules. Right? So again, very important. But you notice now the patterns. There's patterns in the formulas. There's patterns in the structures. There's patterns in the name. And that's why they're grouped together, and they have specific things that they do in the body in this case. All right, so now I think I have to switch to the other computer because I want to do one more iClicker quiz, okay, just to get us practicing here a bit. Let's see, am I on the, because I can't run iClicker from this, so I do need to switch over to the other computer to run the iClicker. All right. So I'm going to start it, and you're being asked here for histidine, is it a neurotransmitter, amino acid, a sugar, or none of the above? Okay, so, and you're welcome to talk to the people around you, and we'll emphasize that more next time, so that you learn to sit with other people and get to know them and discuss your answers. So, in 1 minute or so. Okay, everyone finished? Whoops. Oh, didn't mean to. Forget the next one. Don't worry about the next one. I'm just looking. I just want to display the first one here. All right. So there's some debate here about whether histidine is an amino acid or a neurotransmitter. I don't have a whole lot of time to discuss that, but what's the thinking on what are you looking for to decide that it's an amino acid? What do you see? Go ahead.
You can see the amino group and the attached to the carboxyl group, and
Mm-hmm.
then it's, when you look at the space filling model, it also doesn't, because it's between the neurotransmitter and amino acid, it doesn't have the shape. You have benzene with the self-attached.
All right, so there, in fact, the one below it actually shows the benzene ring with the trail coming off it. So her argument was that she saw both the nitrogen NH2 group and the C with the two bonds to the O and the one bond to the OH on it, and the shape didn't look right to her for the neurotransmitter. But why might you have thought it was a neurotransmitter? There's good reasons for that. Go ahead. ends in INE, right? So that might have clued you in. So that might have been a little bit confusing because they both have amine groups in it. Okay, what else? The structure formula. So go ahead. Has the same type of atoms. So this one is a little bit ambiguous. Histidine is, in fact, an amino acid, but in this case, what I would have been more interested in if I had asked this on an exam was your reasoning. And the reasoning for both of those was really quite good. So, so you want to practice justifying what you're choosing and why you're choosing it. So let me just get to the end. We'll always have wrap-up points. This is a good guide for you. I will also post a study guide for this lecture after it, and I will post the pre-class assignment for the next one. And so you know what to go look at and make sure that you understand. By the way, here's a really good thing for you to do. Steroids, cortisone, progesterone, testosterone. Does it all sound like they might have some similarities? Go look on the web. See what the molecules look like. Convince yourselves that they're all similar. Now, we always like to send you off the first day with a welcome. And what I have here is a hydrogen balloon. We will do lots and lots of demos. I've got my earphones, my earplugs. You might want to hold your ears. So hydrogen is just sitting there. Nothing happens. What do you think is going to happen if I give it a little bit of energy from the flame? Okay, you ready? All right, I'll see you all on Monday.